Jai Hind students, so let's start with the today's class. In our class today, we are going to start with the topic cilia and flagella. So let us see that what is cilia and flagella. Cilia and flagellas are the two appendages, two parts which are responsible for the movement of the organism. Alright, so as they are the flagellated structure, so they helps in the movement or occupying the distance of the organism. For an example, if you see the sperm cell is over here, so this sperm cell is having a long tail. Now as this tail will show the movement, the sperm part, the inner part is also moves and travels its distance, right? So the same way it is also working. So now let us see that what is this cell. Actually from the cell membrane, for example this is the cell membrane of the organism, so from its surface only a cilia will grow somewhat like this, which is quite longer one, and as this will move the body is also going to travel. Apart from the cilia, there are the small structures, appendages like this, and are called as a flagella. The small structures are called as the cilia. These are really very small and help to move, to show the movement, to occupy the distance of the organism. Alright, so now what we are going to see, we are going to see in this structure about the features and the structure of the cilia. So let me show you that what it is, the diagram is actually. As you can see over here, if you will cut this flagella from here, alright, so the circle which is formed over here, that circle is this one. For an example, if this one is a flagella and I had removed from its surface, I had cut it over here, so the circle which is over here, that we are observing or we are studying over here. So let us see that what it is actually. Cell membrane means it arises directly from the cell membrane. Motion, it shows the motion, it helps the organism to cover the distance, to occupy the distance and to travel. Emerge from the body basal, that means it has emerged from the body basal, from the body basal surface only it is occupying its space and working. It is having 9 plus 2 appendage. What is this 9 plus 2? Let us see that first. Okay. So this 9 plus 2 is really very important actually. As in this particular diagram, you can see over here at this place I had made 1 and 2, right? So as you can see these two circles in the inner side which is complete one, this is called as the singlet, as they are single and 2 in number. So there I had mentioned 2 and it is the singlet one I had written. Okay. Next we are having their number 9. So as you can see this 1, 2, 3, 4, 5, 6, 7, 8, 9. Why is extra? Okay, I'll erase the one extra one. Okay, so there is this 9 circle structure which you can see over here. This is 9 in number. So I had mentioned over there 9. Now you can see the structure of this circular structure. This is not actually two different circles. If it would have been two circles, then it would have been like this, right? But it is one circle is like this and the other one is like this. So this is the one complete circle and this is just left. Okay, this is not the complete one circle. So this is the your 9 in number. So this is 9 in number. Now we'll see that how it is made later on. Let us see first that what strokes they are having and what are their different features what they are having. So this one circle is A and this is the B, right? So this A circle which you are watching over here, this is having some extra appendages coming out from its body. So we'll see that also. Okay, let us see the first wall kind of a structure which it is having. This is called PM, that is your plasma membrane. This plasma membrane helps to do the transportation of the materials, fluid materials of it. Then next we are having Nexulin. Nexulin is actually present over here to join these doublets. These two circulars, these circle structure which is having A and B circle is called as actually doublets, which you can see over here. So this is called as a doublet and this one is the singlet. So now to join two doublets, Nexulin is present over here, right? Now Nexulin joins these two doublets and here to join two doublets at this place tectine is present. So tectine is actually a connecting protein which joins two different circles in the doublets. Now next we are having the radial spoke. This radial spoke arises from the A circle and gets attracted towards the central sheath. This one is a central sheath. Alright. So now this central sheath is actually covering for the singlet, and now the radial spoke gets attracted towards this and it joins between central sheath and the doublet. Now next we have seen tectine, doublet, Nexulin, PM, radial spoke, singlet, and now we have to see dynein. Actually, the work of movement gets started from the dynein only. Now what happens, suppose this is the dynein, okay. So on this is a doublet for imagination, and this is the dynein, right. Now this is the another doublet, this is its dynein, this is the another one, and this is its dynein. Now what will happen, this dynein will get tilted towards down. When it will receive the stimulus or any solvent, fluid, solidity fluid, or whatever movement it is receiving, and it is getting influenced by or stimulus it is giving, it will just tilt itself. And when this is going to tilt, this will also tilt, because they all are connected, interconnected, right? So now when this will move like this, the whole 9 doublets will move like this. This will get tilted towards this side, so this kind of a movement will be there. And same with the vice versa. So the movement which is shown by the flagellas or the cilia is just because of the dynein. Okay, so this is really star marked question, which is really very important, that which part helps to the movement in the flagella. So it is always a dynein. Alright, now let us see that what is composition, or what it is composed in the doublet, how it is made, how these doublets are made. Alright, so now let's start this. You must have seen, or you must have studied in your previous topics about the protofilament ring. Okay, so these are actually made up of a protofilament ring, and this protofilament ring is there which makes a complete vertical line for example, this kind of a rectangular structure is there which is made up of a protofilament. So if you will talk about this, that how doublet is made, or what is doublet actually, so doublet is a protofilament ring. Okay, doublet is a protofilament ring. Now what happens in this, a long chain, straight chain kind of a structure is made which is of a protofilament. So this is the protofilament. And now this is straight line of a protofilament. It makes the alpha and beta kind of a tubulin monomer in the 30 numbers. So this kind of a circular structure. Okay, so this is alpha, this is beta, alpha, beta, alpha, beta, and here also alpha, beta, alpha, beta. So that kind of a structure, 30 numbers are made. Okay, so this circular structure which you are seeing, circle, circle, alright. So this circle, circle is actually this your doublet, which is having 30 protofilaments, 30. Okay, so these 30 protofilaments, if you are observing it like this, you will see that this circular structure is having 30 protofilaments like this, and it is making its circle. Alright, so hope now this one is clear to you, that how your doublet is made up of, or how it is made, right? So this much is about your cilia and flagella. Now further we'll see the next topic.